I am going to describe the methods or the processes through which we are isolating DNA. All processes, whether you are taking DNA from plants, you are taking DNA from animals, you are taking DNA from say microbes. So that isolation we can achieve, whether it is plant, whether it is animal, whether it is what we are saying microbial subject, from 2 to 3 hours. The experimental part we are performing 2 to 3 hours. It is job of 2 to 3 hours. Sometimes overnight process is required. But I will provide you such method, little bit modified one, modified through practices. So that is costing about 2 to 3 hours, and in a single day you can isolate DNA from any tissue. Mechanism almost same. In case of say plants or animals, you are having nucleus available. Within nucleus you are having this DNA associated with nucleoproteins called as nucleosome or what is called as chromosomes later on. Or in case of bacteria, where you are lacking mitochondria or nucleus or anything, such type of DNA is just embedded within the membranes. It is also associated with proteins called as nucleoproteins or called as these nucleic acid binding proteins. So your first step in mechanism for isolation of DNA is to rupture cell. So your first point is to clear out to rupture your cell so that nucleus or nucleoproteins will come out. Okay, that can be achieved by various methods. Rupturing can be achieved by using sometimes, in case of what is called as bacteria, you are having peptidoglycan there. You are rupturing its cell wall, that is peptidoglycan, by using some enzymes called as lysozymes. You are having plant cell where you are rupturing cell wall through cellulases or sometimes pectinases. Even sometimes lignin is also associated lignases. So lot of enzymes you can require there for rupturing the cell wall. Not only this, you are having buffer system, because buffer system should be of such pH and concentration where your DNA molecule, the molecule of choice I can say, is stable. You have to keep that in a stable position. So only for that very purpose you are requiring what is called as a buffer of proper pH. So usually tris buffer is preferred in this case, because when you are going to have tris buffer into consideration, its pK is lying within the range of 8, so that we can achieve pH 7 to 8. Second thing, we are requiring some other thing which is rupturing your cell wall or cell membranes then later on. That can be achieved by detergents. Detergents can be partly charged like CTAB. Detergents can be negatively charged like SDS. Detergents can be neutral charged like Triton X100. So any sort of detergent you can utilize. But some specific detergents are utilized for specific purposes. Like in case of plants, you are taking their detergent, usually CTAB, not SDS and other thing, because CTAB, it is a detergent with positive charge, finally ammonium bromide, it is finally hexadecyl, etc., ammonium bromide. That means it is positively charged detergent. It is having role in something, because plant cells are containing lot of carbohydrates, polysaccharides you are knowing. So in order to get rid of such polysaccharides at a particular concentration of sodium chloride and a salt concentration, one thing is precipitated out, another thing is remaining in soluble form, or another one thing is precipitated out, another thing is taken in the soluble form. So that between two, in a DNA molecule, it is remaining in soluble form at a particular salt concentration, and at higher concentration sometimes you are having the carbohydrate in their precipitated form. So you are removing by using this type of detergent, and in a single go it is also taking your molecules of choice, what are nucleic acids, RNA, DNA, because of positive charge and negative charge interactions, it is taken in that association. So there is a mechanism that why we are using CTAP there. And in general, we are using SDS. I can use SDS in case of plant system also, even bacterial system also. This is rupturing cell wall because it is containing one very big hydrophobic chain, long chain, and negatively charged sulfonate groups on another side. So it is having both polar size as well as non-polar nature, called as amphipathic in nature. And this amphipathic nature is capable of disrupting the interactions of the membrane, proteins, etc., as well as the phospholipid membrane, which is associated through biliary system, and that is ruptured by what is called as the long chains of such thing, because hydrophobic interactions are involved there then later on. So rupturing of cell wall, cell membrane is taking place, and after that nucleoproteins are coming out. Nucleoproteins are also, because of the detergent action, they are separated out. Not only this thing, sometimes we are requiring magnesium chloride concentration in order to have membrane, this is what is called an organelle separation, or separation of RNA from nucleoproteins or DNA from nucleoproteins. Finally, what is coming out? We are associated with only one thing, that is DNA. We are only taking that thing into consideration. What is happening to others, we are not bothering about. So your buffer system should contain the proper pH with proper pK and proper ionic concentration, because cell membrane isotonic, what is called a hypotonic nature is to be maintained, so that some water will move out inside cell will be ruptured out because of osmotic tension also. So lot of forces are coming there into play, which are helping in cell disruption. So for that purpose, first point is we are requiring for plants, for bacteria, or for any other system, we are requiring buffer system along with the detergent, so that you are going to have disruption of cells, disruption of what is called as nucleus membrane bound these organelles, and you are getting finally DNA out. DNA will be in solution form. Now what is your choice now? You have to go, you have to remove the what is remaining there, cell debris, whatsoever it may be, some organism even, you are then centrifuging it at a particular what is called as RPM or RCF. So RCF, sometimes in our books, in old and traditional books, are in new books it is RPM, and old it is RCF. In RPM there is no mention of it is only speed. Here it is mention of G later on. You are having sometimes 2000 G, 5000 G, 10,000 G. Are you knowing this thing? It is called a relative centrifugal force. Here you are taking centrifugation with respect to gravitational pull also into consideration, and whatever force you are giving there, that is called a centrifugal force with respect to gravity. But it is only rotation is permanent. It is also related to relative centrifugal force, because there is after mathematical calculations, there is very big formula. You are not capable of remembering that thing because it is very tedious and it is very difficult to remember this thing. So that is 1.119 into terms of 5 RPM whole square into omega square into x, omega काट ले, ये आया इसमें डिवाइड बाय, बस इतना ही वैल्यू आपको यहाँ पर याद रखना है, क्योंकि RPM I have converted from that. So what is one point, that this value you can convert into this value. Only you have to take RPM into consideration. And axis of rotation. Axis of rotation means it is a line of axis about which rotor is moving like this, and you are having here say centrifuge tube. This centrifuge tube is containing matter. That matter is at a distance of x. Depending upon a long axis, you are going to have more centrifugal force there, even in a single machine. So sometimes it is in RCF, sometimes it is written there RPM. So no problem, you can utilize that thing. Nowadays machines are available which are giving their own, their RCF converted into RPM, they are giving that thing. So you are centrifuging it, and you are getting the precipitate containing a lot of other things, and your DNA will be in solution form. Now what is your next step? Single two or three steps. What is next step? Yes, your next step is to get DNA only. That is purification from other components. It is isolation. Whole process is isolation. The second step is now you are releasing DNA, It is in solution, and the solution you have to get DNA out. So there are some other processes which you are taking into consideration. Now this from plants I am saying, say the solution is containing now RNA also. Is it okay? It is containing DNA. So it is containing histones, that's everything, other proteins. So 3 components it is containing maximally now. Because other components you have taken out because of their precipitation, because there is high centrifugal values, S values, these are taken out by centrifugation. But it is containing a lot of other things, but these things are now in maximum. From these 3 and other things, you have to get which thing out? DNA. Your choice is only DNA, not RNA. So there, from this step, I will utilize different procedures, different processes for isolation. It is your choice. Some people are, in case of plant system, overnight procedure, they are taking this, now it is aqua solution. Say it is now 1 ml total coming out, 2 ml coming out, what is remaining? Supernatant, it is called as, say it is 1 ml or?
2 ml.
You have to keep that, you have to measure that thing. Now to this 1 ml, you have to add equal volume of PCI. What does it reflect? Phenol chloroform isoamyl alcohol. See, PCI, phenol chloroform isoamyl alcohol in the ratio of 25:24:1. A point very important. Keep it this point. It is very important point. You have to now add what thing? Equal volume of PCI. Equal volume. Not necessarily too much. Sometimes you can take a little, but in order to have a good value of DNA, purified DNA, and good quantity of DNA, you can utilize equal volume of what? PCI. Now question is what PCI is? I will represent here. It is phenol, it is chloroform, it is isoamyl alcohol. Do you know what is their role? What is their function? You should be having this point in mind. It is very crucial and important in case of DNA, RNA, anything as isolation. What is the role of this now? Phenol? Anyone? No. No. Phenol is called as carbolic acid. Have you heard about it?
Yes.
It is only one containing OH group which is acid. Chemistry people must be knowing very well because of the conjugation system. So it is acid, carbolic acid. But we are saturating it with buffer. It should not be as phenol, it should be saturated phenol. Keep it in mind. Because when it is acidic in nature, it will not differentiate, then it will disrupt pH of solution, it will not then distinguish between RNA and DNA. Because RNA and DNA is having difference only in solubility, little bit solubility difference is there. So because of its hydrophobic, that benzene ring system, I can say like this, it is phenol. This phenol is like this, but it should not be as a phenol, it should be saturated phenol. When it is saturated with this same buffer twist, it is coming in the form of coinoid-like structure, that phenolate structure, phenolate. Because it is forming salt at a pH of 7, 7.4. This phenol is having no doubt now hydrophobic ring system. This phenol is precipitating proteins only, because proteins are containing themselves phenylalanine, tyrosine, tryptophan. Aromatic amino acids. Because of their presence of aromatic amino acids, the precipitation, hydrophobic interactions are taking place, precipitation is taking place. Proteins are precipitated in phenolic form, in phenol phase. Now chloroform and isoamyl alcohol, these are differentiating, taking your solution RNA and DNA. DNA will be now in aqueous form. It is separating it into aqueous form. Aqueous is already buffer, etc. And chloroform, which is there, it is taking RNA and other things. Chloroform is taking RNA and other things, and DNA is going to aqueous system. But isoamyl alcohol is also added. This is helping in separation of two. Number second, it is preventing frothing. If frothing is there, then overall mixing is again there. No separation will be then. Yeah. So two things it is doing. One, it is creating organic phase, allowing RNA to go there, and it is creating the preventing frothing of the system, so that you are not going to have mixing up of the system again. Then what is the fun? Are you getting my point? So when you are taking this PCI, you are adding equal volume of PCI, then you have to go like this. Yeah, you are having that shocking this rack, or what is called as that shaking machine, or what is called as shaker. You are going to shake it, or you are going to take it like this slowly, invert like this for time being. Okay? For 10, 30, 40, 50 minutes. It is up to you. You can take even half an hour. So that proper separation is taking place. Then you can go there. You will take it 30,000 RPM. 13,000 RPM. You can spin for 1 minute only. There will be separation of the layers. Layer separation will be there because 3 compounds are now there. One is the denatured proteins, that is in phenolic phase. Second is RNA going in organic phase. Topmost will be now your aqueous phase. So how many phases you are going to achieve there? One will be denatured proteins like this, bulky one. Second will be your organic phase containing your RNA, etc. Any other thing also. It can be lipid also. Because chloroform is not only for RNA, it is also dissolving lipids if they are present there. And the last one is containing your DNA. Now DNA is also containing a lot of polar molecules associated. DNA is also containing a lot of polar molecules associated molecules. They can interfere still. But from such huge number of molecules, we are going to now have only DNA. यहाँ तक RNA protein थोड़ा खत्म होने लगा. लेकिन in one go, you cannot achieve 100% purification as you are expecting. No. You can take this aqueous phase once again. You can add again PCI. Yeah, only CI now. No need of P up. Only CI. Sometimes PCI is better. So that again you can shake it for half an hour. You can get then centrifugation. You can get 3 layers out. Are you getting my point? So again you will achieve what phase later on? अगर अभी भी सेटिस्फाई नहीं है तो तीसरी बार कर लो, 3 तक आपका ये है. तो after that, whatever is remaining in aqueous phase, that is containing what DNA and all other molecules which are not going in aqueous organic phase, which have not been precipitated out by the phenol. So that thing containing DNA and other things, now we are going to have only DNA isolation. And DNA purification, you can say DNA purification even. So what is to be done? This aqueous phase part, how much it is? See, it may be 1 ml, it may be 0.8 ml, it may be even 1.2 ml. You can take that out. You can add direct phenol to this alcohol to it, or isopropanol to it. Isopropanol, yeah, ethanol. Usually people are using ethanol. See, मैं इसका मैकेनिज्म बोलूँगा, लेकिन इससे पहले एक चीज़, in order to achieve better precipitation, good precipitation of DNA, you are adding something more. Anyone? आप करते रहते हो बहुत बार. Anyone? Sometimes you are adding NaCl, sometimes you are adding 3 molar sodium acetate. 3 molar sodium acetate you are making. You are adding, if it is 1 ml, now aqueous phase, you are adding up to 100. That is 0.3. And if it is 3 molar, you are adding only 0.1 to 1. It is now 0.3. Overall, it should be 0.3. But concentrated form, it should be 3 molar sodium. See what is happening. Hey, what is happening? In aqueous solution, you are adding now DNA, lot of other things. When you are adding salt, sodium acetate formula is CH3CONA. When it is in aqueous form, it will be in the little dissociated, it will be in the form of this. Are you getting my point? Yes, so you must be knowing DNA is itself what charge? Negative charge molecules repel each other. So they are not they will repel each other, and when you are going to achieve precipitation, they will move apart. Precipitation is less. Yeah, we will not achieve good precipitation. Now, this sodium is covering the part of negative charge, making it neutralizing it, so that shielding it. You can say shielding it, charge shielding, but no, we can say neutralizing it. So shielding of charge. Then what is happening? No charge. Then yeah, charge less we can say, but it is not chargeless. So that we are adding then ethanol, ETHANOL, C2H5OH, but it should be chilled. Chilled, you know why? Chilled means deep freezer से निकाला हुआ 20 माइनस पर, ये 20 तक नहीं हो जाता है, what is called as ice, ये liquid रहता है, तो deep freeze में लेते हैं. तो what is happening? What will happen? When you are adding ethanol, ethanol is having OH groups. The DNA is soluble because of water content available for DNA within the system, too much solubility. And when you are trying to abstract water from DNA, to get abstraction of water from DNA, then what is happening? Because you must be knowing in DNA structure, the hydrophobic bases are interior to the structure. Only phosphate groups negative are outside the system. Negative phosphate groups are making water interaction through hydrogen bonding, and solubility is maintained. But when you are abstracting that water by alcohol, because alcohol is more powerful than that structure which is inside, then water is going towards alcohol, because alcohol is having higher solubility for water, and water is removed too much, a little bit I can say, then hydrophobic patches are coming out. And when hydrophobic patches are coming out, they are collapsing with each other. Number one, when collapsing of hydrophobic patches, what is called as the bases, adding one cytosine, whatsoever it may be inside, it is coming out. The base interaction is coming into play, and collapsing of DNA is taking place, that is called as precipitation. Second thing, for repulsion, you are removing negative charge, so no repulsion. Another force is added. They are coming more together. And third thing, that is you are using very, very low temperature alcohol. Temperature is creating 1/2 mv square. अगर पता होगा, अगर नहीं 3/2 RT. क्योंकि motion is, temperature is decreased, motion of molecule is decreasing. When motion of molecule is decreasing, they are coming close to each other, and collapsing is again added. Third thing, your DNA molecule is vulnerable for alcohol at higher temperature, because it is seeping deep into the base structures. It is preventing that thing, because at low temperature the denaturation process is to some extent prevented, controlled. इतना आया समझ? ये बहुत important point था इसमें. And what is happening? After addition of this alcohol, you are going to have precipitation of DNA. Some people can see it by their eyes, the cotton-like structures, cotton-like that fibers produced there. You can directly take that out by cutting the nose of what tip and taking it out, keeping it in a separate tube. It will be purified one, because others will be within solution again, some components. Or you are allowing it to precipitate out, then you are centrifuging it at 13,000 or 10,000 RPM for 10 minutes. Precipitate will settle down. That will be DNA. Maybe some other molecules sometimes, not 100% I can say, but DNA will be there. If you are taking only by the tip, only DNA, that will be only Khal's DNA. But you are precipitating it out, it will be at the bottom, that will be DNA. But maybe something acid or salt भी थोड़ा होगा या नहीं? तो precipitate you are taking there, then you are washing this precipitate. When you are leaving now alcohol out, there will be in the tube precipitate at the bottom. This precipitate may be containing some salt ions. You are going to remove salt ions, but you have to keep in mind DNA should not be in dissolved form. It should be as precipitate, and only salt and other small molecules should come out. Then you are adding 70% alcohol. इसलिए 70% alcohol उससे wash करते हैं. 70% means 70-30. Yes, sir. 70 alcohol का 30 मिलिक्यू वाटर का. गोबरा समझ आया? तो it is just थर्टिकल एक्चुअली हम ऐसे नहीं बनाते हैं. People from chemistry must be knowing this thing. Actually solution ऐसे नहीं बताते 70 मिलाया 30 मिलाया. It is not the way, because no solution is real solution, ideal solution. So there is sometimes contraction because of hydrogen bonding, volume is then becoming less. So it is total should be 1. Total should be 100 ml. Whatsoever it may be, no hard and fast, not too much deviations, because we are requiring it from 65 to 75%. 30% water which is present within the system, that is removing salts, and 70% alcohol is keeping this DNA intact as a precipitate. Clear hua? And then you can wash it number of times. Finally DNA precipitate is at the bottom. You can take this tube, you can place it in near dryer, hair dryer. You can keep it for overnight, inverted. So alcohol will be evaporated out. You can take that precipitate now. You can dissolve it in 50 to 100, 30 to 100 microliter of distilled water. Distilled water मत कर लो यहाँ पर स्पेशल वाटर आता है, मिलिक्यो वाटर. अगर बहुत देर के लिए फिर ये रखना है तो टी बफर का मतलब ट्रिस, EDTA, because EDTA is preventing DNA's action. If may be present, DNA's action. Even sometimes you are keeping tube open, bacteria or something is going there, that is producing DNAs. DNA will be taken care of. It will be disrupted. But TE buffer is preventing. T is tris, it is preventing buffer because pH, it is giving proper pH. And E is EDTA, it is chelating that magnesium ion is not allowing DNAs to have action. So it will be for 6, 7, even 2 years.